Welcome everyone. Thank you so much for coming to this BrainMap seminar. As always, this seminar series is co-sponsored by the P41 Funded Center for Mesoscale Mapping, housed here in Martinos Center. And today we are delighted to have Dr. Kevin J. Chung. He's American Heart Association Postdoctoral Fellow in the Department of Radiology at UC Davis Health, supervised by Drs. Guabao Yang and Simon Cherry. Kevin's work has been recognized with several early career awards, including ISCBFM Niels Listen Award at 2025 Brain and BrainPad Conference. Kevin will give us a highly anticipated talk today named Total PET across scale system. Thank you so much, Kevin, and welcome.
Okay. Thank you very much for the invitation to speak at this special BrainMap seminar. I'm happy to be representing the Explorer team at UC Davis, of course, where total body PET was first developed, and where I've had the privilege of working over the past 2.5 years. So I'll showcase some of the work that we've been doing, taking advantage of the unique capabilities of total body PET, namely in terms of its high sensitivity, as well as the ability to dynamically image the whole body. And hopefully to try and get people excited about this new technology that will soon be coming to the Martinos Center. So there are two key features of PET that really stand out that I want to highlight for this presentation. And the first is that it's a highly sensitive platform for studying molecular processes in the human body. And this is really enabled by the many radio tracers that we have access to that specifically target molecular or physiological processes. Of course, the most widely used PET tracer in clinical practice is FDG, which gives us a nice measure of glucose metabolism. And we have and PET imaging of blood flow is generally considered the gold standard way of measuring blood flow in the human body. We can do this using flow-specific radio tracers like O15 water or carb-11 butanol. And we have this large library of radio tracers with which we can study the body's biochemistry, and all that to boot, we can also spatially map its biodistribution as well. And the second key feature of PET is that it's inherently a whole body imaging modality. So once we inject the radio tracer into the patient, it circulates throughout the whole body and distributes, emitting detectable radiation. So really the question is whether we can place PET detectors over a particular region of the body to actually image it. And so our conventional PET scanners span about 15 to 30 centimeters, and we can perform whole body imaging by sort of sequentially feeding the patient through the scanner and stitching together those resulting images to form a whole body picture. We typically do this at a single time point after radio tracer injection. We call this static imaging. And this gives us a single sort of gross measure of tracer uptake in the body, which we generally consider semi-quantitative because it's not specific to the molecular process or any specific molecular process, but rather a gross measure. So for better quantification, we can perform dynamic imaging where we continuously image a portion of the body and measure the change in activity concentration over time. This is called, or these are called time activity curves. And by applying tracer kinetic models, which mathematically describe how we expect these tracers to distribute and interact with tissue, we can get better quantification of these molecular processes. For example, we can quantify these kinetic rate constants K1 to K4. K1 represents, say, the extraction of tracer from blood into tissue. K2 is that reverse process, clearance back into blood. And K3 and K4 might represent the association and dissociation rate constants, or how the tracer is interacting with its molecular target. So now we have better quantification of these underlying molecular processes. And if we can reliably measure more than one of these parameters, then we have the potential to perform multi-parametric imaging from a single PET scan. But now we've sort of lost this ability in general to image the whole body. And these time activity curves that I'm showing are somewhat misleading because in practice, we can have quite noisy time activity curves. So we have poor signal to noise ratio. And how do we get an arterial input function, which tells us, or tells the model, how much tracer is actually being inputted into the tissue because of the poor spatial resolution of PET systems. So how can we sort of get the best of both worlds in terms of imaging the whole body, as well as being able to quantify these molecular processes? And really the challenge is that with conventional PET systems, we only have a short span of PET detectors that cover a fraction of the body at one time, despite detectable signal being emitted from throughout the whole body. So these oblique lines of responses are completely undetected, as well as any activity that's being emitted from outside of the axial field of view. And it's been shown that, in fact, less than 1% of the total available signal is actually detected by our conventional PET systems. And so there's a simple but radical idea that was proposed to tackle these challenges, and that is to cover the entire body with PET detectors. And this concept is called total body PET imaging. This results in a huge boost in signal sensitivity, about 40 times at the total body level and about 5 times at the single organ level. And importantly, for a kinetic modeler like myself, we now have for the first time a device that can dynamically image the entire body at the same time. So this total body PET CT system was developed by a collaboration with the Explorer Consortium, which was led by Drs. Ramsey Badawi and Simon Cherry at UC Davis, funded by a NIH Transformative R01, and was manufactured by United Imaging Healthcare, who also commercialized it and took it through the FDA clearance process. This picture on the left is the world's first U-Explorer total body PET CT system that was installed at our Explorer Molecular Imaging Center in Sacramento, California. And of course, the dominant sort of characteristic of the scanner is its nearly 2-meter long axial field of view in terms of the PET components. So this picture on the left is sort of what we use for these official presentation purposes, but I don't think it does it quite justice to the length of the scanner. So this is a picture of me lying down next to the scanner. And I'm not a particularly tall person, but I hope you can appreciate sort of the scale of the system. So there's an integrated CT scanner at the front for attenuation correction and anatomical localization. And since the first one was installed in around 2018, 2019, there are about 50 such long axial field of view PET systems worldwide, with one soon to come at the Martinos Center. So what can we do with 40 times more signal? Well, given the order of magnitude greater sensitivity, we have a lot greater flexibility in terms of our imaging protocols. We can choose to simply image better, get unprecedented high quality molecular images of the whole human body, or you can reduce the injected activity to reduce radiation dose by at least tenfold, or we can stretch the number of half-lives that we image beyond to start studying slow biological processes using longer-lived radioisotopes. And you can simply image faster, obtain diagnostic quality images in less than 2 minutes, opening up new opportunities for patients who otherwise could not comply with longer scan protocols. My research specifically focuses on this new ability to image the entire body at the same time dynamically. And we've been pushing the boundaries of what we can do with this technology to perform fast, high temporal resolution dynamic imaging. So this is an example of a total body dynamic PET scan where we've chopped up these dynamic frames into 1-second frames, which can nicely show the initial injection and first pass vascular circulation of these radio tracers. And so we have simultaneous dynamic imaging of all major organs from which we can obtain these nice high temporal resolution time activity curves, including the aorta, which can be used as a image-derived whole blood arterial input function for kinetic modeling. So my work specifically is focused on answering the question of how do we actually analyze these images to extract as much information as possible to take advantage of all the rich information that's contained in these total body dynamic data sets. So in this presentation, I'll give 3 examples of how we've been taking advantage of the unique capabilities of total body PET, from quantifying blood flow across the entire body, not using a flow radio tracer, but rather non-flow radio tracers like FDG to open the possibility for total body multi-parametric imaging. Then we'll zoom into the molecular level and start studying blood-brain barrier permeability of molecular radio tracers and putting these pieces together to see how the brain and body might interact with one another in a representative systemic disease. So I'll start with the total body blood flow imaging project. So the conventional way in which we perform our PET studies is we generally have a radio tracer that gives us a really nice measure of a specific molecular process. So FDG is commonly used to assess glucose metabolism, or we have flow radio tracers like O15 water or carbon-11 butanol that give us nice measures of blood flow. But these flow radio tracers have short radionuclide half-lives and are generally quite challenging to work with. And so given that we have this new opportunity to rapidly sample the vascular circulation of our radio tracers, here we investigated the possibility of performing multi-parametric imaging with FDG by performing quantitative blood flow imaging of the whole body by analyzing this initial vascular transit of these radio tracers. So the challenge is that with conventional PET kinetic modeling, we use compartment models to measure blood flow, and we really focus on this K1 parameter. K1 is the blood to tissue transport rate, or how rapidly a tracer gets extracted from blood into tissue. And K1 is a good approximation of blood flow when the tracer extraction fraction is nearly 100%. And this is the case for flow-specific radio tracers like O15 water or carbon-11 butanol that are nearly completely extracted throughout the whole body. But this is not the case for FDG, which has limited extraction in certain organs, like in the brain and the kidneys, where extraction is about 20 to 40%. And extraction fraction is also not uniform across different tissues as well. So we can't use this conventional method for total body blood flow imaging with FDG. So what we propose is to directly model intravascular transit of our radio tracers to quantify blood flow. This normally occurs on the order of seconds. The mean vascular transit time in the brain is about 3 to 7 seconds. And as a result, we really do need this high temporal resolution imaging capability, which is now made available with total body PET. And specifically, we use the adiabatic approximation to the tissue homogeneity model, or the AATH model, which provides us this nice closed form time domain impulse response function to a more complicated distributed kinetic model described by a system of partial differential equations as opposed to ordinary differential equations for our compartment models. But importantly, the AATH model allows direct estimation of blood flow F, which is enabled by this distinct modeling of the vascular phase of tracer transit. And that's the time it takes for the tracer to go from the arterial end to the venous end in volume of tissue. And this is in contrast to our compartment models, which is missing that vascular phase. And as a result, we can't directly measure blood flow without a flow radio tracer. So to validate this approach, we conducted a study in which we scanned patients on our total body PET system using a representative flow tracer, butanol, and a representative non-flow tracer, FDG. And these are parametric images of blood flow across the body. And I hope you can appreciate the similarity between these two parametric images, both visually and in quantitative range throughout the whole body. And our pilot study consisted of 6 Yep.
You see these venous the structures on the FDG brain, for example, central sinus,
Yep.
Why on that rather than the flow tracer?
Right. So with the flow tracer, it has high extraction. Nearly 100% of it is extracted by brain tissue. So I think what's happening is it appears that there's very little venous concentration of butanol because it's completely extracted into the brain. And maybe the vein and the tissue is in equilibrium between one another. Yes, yeah. I think it's just in terms of how the signal is weighted. It's more like the tissue and vein is in equilibrium. And so it appears as though there's not much tracer in the vein.
And maybe why does it show up on the flow map of FDG?
Right. In FDG, it's only about 20 to 40% extracted in the brain. So there's quite distinct venous clearance of the FDG tracer.
Doesn't suggest there's something missing in the model.
Possibly, yes. Yeah. Could be that the model is incomplete. Yeah. So in our pilot study, we studied 6 participants scanned with both radio tracers, and here we're showing strong correlation between butanol measures of flow as well as FDG across these 10 different regions of interest. Our blind Altman analysis suggests that maybe we're underestimating blood flow with FDG a little bit, but overall we're doing quite well in terms of measuring blood flow throughout this wide range of blood flow values and across different tissues. And so in principle, we should be able to apply this methodology across radio tracers, again, using the principle that our tracers are intravenously delivered and circulate throughout the vascular system. So this is an example of applying our method across these 5 different radio tracers, showing that our blood flow parametric images appear rather comparable across these different tracers. So what this means is that in addition to assessing the molecular process that is targeted by the radio tracer, we can also, by studying the early dynamics of tracer transport, possibly obtain a quantitative measure of blood flow, as well as any intermediate transport processes like FDGK1 or glucose transport. Each of these parameters represent a distinct physiological parameter, and image contrast also appears to be different as well. And so we think there's a lot of potential in more comprehensively characterizing conditions using a single FDG PET scan. So now I'll zoom into the molecular level to study blood-brain barrier permeability of PET tracers. So the blood-brain barrier enables highly selective transport of molecules from blood to the brain. It's lined with tightly joined endothelial cells and wrapped around by astrocyte emphy, among other mural cells, which collectively prevent molecules from just leaking from blood into the brain. And instead, many molecules have to be specially transported through these transporter systems. For example, glucose is imported into the brain through glucose transporter 1 or GLUT1. So the blood-brain barrier facilitates nutritive transport while filtering out potential toxins. A dysfunctional blood-brain barrier is one where there's a loss of this selective or normal molecular transport. And we observe this BBB dysfunction in many disorders, including stroke, brain tumors, Alzheimer's disease, among other neurological and systemic conditions. We typically associate blood-brain barrier dysfunction with BBB breakdown, leading to an increase in blood-brain barrier permeability, where molecules can just start to leak from blood into the brain. And we have established methods to actually assess this in humans using, say, DCE MRI or CT perfusion. But how can we more specifically assess these transport mechanisms expressed at the BBB? We hypothesize that a PET imaging method of measuring blood-brain barrier permeability of these molecular radio tracers might help us probe these systems with greater specificity. But the problem, again, is that measuring blood-brain barrier permeability with PET is challenging by conventional means. Again, we're using compartment modeling, using the conventional method to estimate this K1 parameter, where K1 is the product of cerebral blood flow and the tracer's extraction fraction. By the classical Rankine-Krohn equation, the extraction fraction is related to the ratio of the permeability of that tracer relative to cerebral blood flow. And we can rearrange this equation to isolate for permeability in terms of blood flow and the K1 of the tracer whose permeability we're interested in. But again, this presents several challenges because to measure blood flow and K1, we typically need 2 PET scans, one using the tracer whose permeability we're interested in to measure K1, and another to measure cerebral blood flow, again, using these flow radio tracers like O15 water or carbapenem butanol, which I've mentioned have some challenges due to their short half-lives, not to mention that performing multi-tracer PET studies is also not trivial. And in addition, for brain kinetic modeling with our conventional PET systems, how do we get a reliable arterial input function? Due to the relatively poor spatial resolution of PET, it's not easy to obtain a reliable image-derived input function. And as a result, we often perform invasive arterial sampling to get the arterial input function. And so here, now that we have this ability to measure cerebral blood flow based on high temporal resolution early dynamic imaging, here we investigated whether we can measure blood-brain barrier permeability using a single total body dynamic PET scan. So here we studied 4 different PET tracers, which we expected to have very different levels of blood-brain barrier permeability, going from low permeability to very high permeability based on prior studies. Now we have, with our method, we have the ability to measure cerebral blood flow using each of these different tracers. Here, these are 4 different participants, each scanned with for these 4 different tracers. And we don't expect their cerebral blood flow images to look exactly the same, but at least comparable, which is sort of what we see here. And we also have the ability to measure each tracer's K1, which span from low to very high from left to right as expected. So now that we have this ability to measure at a voxel by voxel basis the cerebral blood flow and K1 for each tracer, we can generate these parametric images of blood-brain barrier permeability for our molecular PET tracers. And here, these 3D renders show quickly at a glance that indeed blood-brain barrier permeability can differ quite substantially across different tracers. Looking at more traditional cross-sectional views, here on the bottom are whole brain permeability averages across 5 different individuals for each tracer. Starting with flucyclovine, we see that sort of as expected, we have very low permeability throughout the entire brain parenchyma. We see some hotspots here in the middle, and this actually is not the blood-brain barrier, but rather the blood-CSF barrier at the choroid plexus, which we know is inherently more permeable than the BBB. For FDG, we see higher permeability in florbetaben, a bit higher than that. And we start to see some gray to white matter contrast. And maybe this is because the gray matter has a higher density of capillaries compared to the white matter. And carbon-11 butanol is this flow radiotracer that's is a lipophilic alcohol and can just freely diffuse across the blood-brain barrier, and as a result, has very high permeability throughout the brain parenchyma. Yep.
Just 1 second. From your kinetic analysis, in addition to blood flow, can you measure blood volume?
Yes. Using the transit time and the cerebral blood flow, we can measure blood volume as well.
And then how could you use that to basically measure the permeability surface area of a given vascular area, rather than its average, which is then weighted by the density of the vessels? Actually, not that measure the permeability surface area of vascular endothelium.
Right. So your question is, can we isolate the permeability itself as opposed to the permeability surface area product, given that we can measure blood volume? I think that's an interesting direction we could look. I think you need to go a little bit further into the details, because I guess our blood volume measurement would be the total blood volume in the vascular space in a tissue, whereas the permeability occurs at the capillary level. So can we disentangle the capillary blood volume to obtain the surface area, and then to disentangle the permeability? I think that's another step that we have to.
Some model of what the vascular bed looks like.
Right.
We actually have pretty good vascular architecture models at this point being developed. So.
That could be an interesting direction. Yeah.
Do you mind just going back to the blood flow? So would you expect them, there's no probability is very low, cyclovine actually lead us right to butanol?
So flucyclovine has the lowest extraction fraction, about 5%, and butanol is quite high, nearly 100%.
Right, but so neither one of them is confounded.
I think in actual practice, when it comes to like the noise of your signal, butanol has much higher extraction, therefore has a very nice signal in the brain. So the images are quite not noisy, whereas flucyclovine has low brain uptake. So the time activity curves are a little bit noisier than that of butanol.
You wouldn't think that's true of the first pass?
First pass, no, the SUVs are quite a bit higher for butanol than for flucyclovine. Just because there's, it's completely uptaken in the brain.
But of course, in a first pass, it's all there. Right? That's transit.
Right. I guess it's the clearance that's slower for butanol. Yeah. So your overall signal is higher. And also your blood flow measure is quite strongly correlated with your K1 because the extraction fraction is very high. Yes. Yeah.
But Peter's point is I don't know if it's subjects, but they are more closely matched at the extremes where their extraction fraction is, it's either very high or very low. It's
Yep. So next we asked, how do we actually know if our permeability measures are quantitatively accurate? We hypothesized that that would be the case if our cerebral blood flow measures were also quantitative. So here we studied 15 participants scanned with both butanol and FDG, and we're showing that across these 5 different regions of interest that we have rather strong correlation between our butanol and FDG measures of cerebral blood flow. So I think taking a step back a little bit, I think this is a different way of thinking about blood-brain barrier permeability, because we're very used to thinking about permeability in terms of these intravascular contrast agents that we typically use for DC MRI or CT perfusion, which normally stay within an intact blood-brain barrier, but start to light the brain when there's blood-brain barrier leakage. And previous work has quantified this process to be on the order of 10 to the negative 3 milliliters per minute per cubic centimeter. But now with our PET method, we can start to probe the blood-brain barrier permeability of our molecular radiotracers and the transport associated with their transport systems. So flucyclamine is this amino acid analog, which we think crosses the blood-brain barrier through amino acid transporter LAT1. And here we've quantified this on the order of 10 to the negative 2 milliliters per minute per cubic centimeter. FDG is by facilitated diffusion through glucose transporter 1 with a permeability on the order of 10 to the negative 1. And florbetaben and carbalambutanol are lipophilic and can just simply diffuse across the blood-brain barrier with higher levels of permeability. So now we have the ability to measure blood-brain barrier permeability across these 4 orders of magnitude, as well as through their molecule-specific transport mechanism. So now we'll put these pieces together to study the brain in a representative systemic disease. And here we're looking at metabolic dysfunction-associated steatotic liver disease, or MASLD, formerly known as non-alcoholic fatty liver disease, which is one of the most common forms of chronic liver disease worldwide. And it's characterized by a spectrum of liver disease, starting with fat deposition in the liver, potentially progressing to liver inflammation or MASH, and potentially progressing to more severe forms of advanced fibrosis that's potentially irreversible. So there are observational studies suggesting that MASLD may be linked with cognitive impairment and dementia, more preclinical studies suggesting or pinpointing MASH as a more specific risk factor. But of course, it's challenging to study how the brain and the liver are interconnected using our conventional imaging tools, simply because most of these tools are designed to look at one part of the body at one particular time. But now with total body PET, we have this potential to start looking outside of the main organ that we're initially interested in. So we have this ongoing study that's aimed at developing a dynamic FDG PET marker of liver inflammation. But since we scan these patients in our total body PET scanner, we also have access to their brains as well. And so we applied our multi-parametric FDG brain imaging method to study whether blood-brain barrier function and brain hemodynamics are affected in patients with MASH. And what we found was that this very interesting trend that patients with more severe forms of liver inflammation, as graded by biopsy, we found that they had lower blood-brain barrier permeability to FDG compared to those with mild liver inflammation and age-matched controls. And this was a very interesting trend because at least I typically associate blood-brain barrier dysfunction with an increase in permeability. Again, thinking back to our DC MRI measures of blood-brain barrier permeability, which indicate sort of the leakage of the BBB. But now, when we're starting to probe BBB using FDG, we found that the permeability can decrease. And actually, this is kind of intuitive because it means that glucose is having a harder time in entering the brain. But this is just a different way of thinking about blood-brain barrier permeability.
And were the trends in the CMR glucose function disease?
In CMR glucose.
In the brain, yeah, several rare.
Yes.
As opposed to the permeability.
I think so far we've just looked at the SUV, which did not show significant differences between these different groups.
But you could estimate.
Yes, yeah, we do have the potential to estimate it. We just haven't looked at that yet. So our regional analysis supported our imaging findings, again, showing that there were significant differences between our controls versus our severe liver inflammation groups. We also found that K1, or glucose transport, also had a similar trend. But interestingly, cerebral blood flow is not significantly different across these groups. So maybe this impairment of glucose transport might be more related to the BBB as opposed to a change in cerebral blood flow. But importantly, using our method, we can now potentially disentangle these different contributions, whereas with the standard compartment model, we could only get a measure of K1. So
what's the magnitude of, say, the blood flow change, extremes versus the magnitude of, say, the PS change?
Yeah, so one sort of confounding factor is that our blood flow measurements are generally have a bit more variability than our permeability measures. So we also see greater spread here. Could be physiological, could be also due to our measurement method. So that's probably that could be one reason why that we don't see significant differences here.
Yeah, that's kind of what the curves look very. I mean, the bars look very similar. The noise levels are different. And I'm wondering if, rather than the conclusion that blood flow is constant, PS changes, the conclusion is one measurement is noisier than the other. The trend is the same.
Right. Yeah. Yeah. So when we're thinking about cognitive impairment in MASH, maybe we should be thinking about blood-brain barrier permeability as well as the hemodynamics of the brain as well. So I hope I've showcased some of the capabilities and potential of total body PET, where we've used this technology to measure blood flow across the entire body, not using a flow tracer, but rather by analyzing the vascular circulation of non-flow tracers like FDG. We have also used this to measure blood-brain barrier permeability at the molecular level using our molecular PET tracers. We've also shown the potential to use this technology for multi-parametric brain imaging in a representative systemic disease. And so I think we're really only scratching the surface of what we can do with this technology. And it's a promising platform to study the human body as an ensemble of interconnected systems as opposed to a set just isolated organs. So what's next? I think our goal as a field should be to try and use the unique capabilities of the scanner in terms of leveraging its high sensitivity for unique organ-specific applications, for example, our blood-brain barrier permeability method, and also to take advantage of the extended axial field of view to get a new view on brain-body interconnections. And also realizing that PET is not the only way that you can do this. By integrating this with other imaging and data modalities, we could potentially paint a more holistic picture of what's going on in the whole human body. So one application of our method that could be interesting is in Alzheimer's disease, where we can better characterize the role of blood-brain barrier dysfunction in the disease pathology. Of course, this is the ATN classification, where the X is this variable that includes these potential factors to the disease pathology, one of which is the blood-brain barrier that has been implicated. And so there's a long history of studying the blood-brain barrier in these patients, with DC MRI suggesting that the BBB may become leakier in AD compared to controls. But what's going on at the molecular level? At least in aging, we've shown that there appears to be a trend in decreased FDG permeability with greater age. And there's literature suggesting that there's impaired glucose transport in these participants. So maybe we can disentangle some of these effects relative to permeability versus cerebral blood flow.
Yeah, saying that you also see a similar trend. Which would suggest that serves.
Right.
So it's a B or it's a BS.
Yes, yeah. And one of the key applications that we're interested in for our FDG blood flow imaging method is its application for the myocardium, where a key diagnostic marker of a viable myocardium is this mismatch between having reduced myocardial blood flow and increased glucose metabolism. Again, the same pattern. It's challenging to obtain this measure of viability because you need two PET scans, one using a cardiac flow tracer and another using FDG. But now maybe we can do this with a single FDG PET scan. And this is an example of a clinical myocardial blood flow scan 9 months after their revascularization, suggesting there seems to be some agreement in our myocardial blood flow estimates. And in cardiac PET, we rarely perform. Yep.
I had a question. So thanks for showing the heart of the aortic flow. Any chance to look into does it vary for at the different glucose level while you inject the FDG, because just given the unique physiology of the heart of the
So,
so like the patient's glucose level, does it change? I mean, like have you tried out different patients for given like you using FDG to measure the MBF?
Oh, does MBF
Yeah,
depend
the
on?
patient I know it's a patient, so they probably most fasted for 6 hours, 8
Right.
hours. So have you tried for patients like a normal diet and at different glucose level
So
while injecting that FDG?
we haven't looked at patients who have not fasted. It's just not in our current protocol, even for research. I'm not sure if physiologically there's a link between myocardial blood flow and blood glucose level.
If we have a same subject that being fasted and not fasted,
Mhm.
and then using FDG and the 2SC.
Right. I'm not sure how that might affect myocardial blood flow. I feel like it would be less effective than, say, glucose metabolism.
So,
yeah.
I'm just wanting to
Yeah.
see that using your method.
Yeah.
It's kind of pretty sensitive to all different. We use a lot of noise.
Right.
Yeah, I think it would be a very good way to test your model, but
Yeah.
just don't expect blood flow to change. Expect uptake to change
Right.
quite a bit.
Yeah, exactly.
Right. Yeah. It's a great point. Yep. So in cardiac PET imaging, we rarely assess myocardial blood flow just at rest. We also look at it under a stress condition. We often do this using a vasodilator to see how well the blood vessels can dilate to increase myocardial blood flow. But it turns out these agents don't only cause a change in the myocardium, but also throughout other parts of the body as well. So prior work at UC Davis has shown that using a flow tracer, carbon-11 butanol, we can quantify rest and stress blood flow across the body, and it even causes blood flow changes in the brain. And so for our next question is whether we can do this with FDG, a common reviewer question, whether we can measure blood flow if we perturb the vascular system. And what that means is that if we can measure blood flow using FDG at stress, we can also get these measures of flow reserves, or the ratio of blood flow at stress versus rest. And this can be a measure of microvascular function, how well the blood vessels can dilate to the stimuli. And so an interesting research direction that I'm interested in is studying the relationship between microvascular function across multiple organs. And I think this is a unique application of total body PET to look at these dynamic stresses across multiple organs at one time. And this is one example of a broader paradigm of trying to think of how do we stimulate the body and assess the coordinated or discoordinated response across different organs. And of course, PET is not the only way you can do this. Of course, I'd be remiss if I didn't mention that you could do this with MRI as well. The huge advantage being that we're free of ionizing radiation, and therefore you can just sequentially image multiple parts of the body for multi-organ imaging. And this has been adopted in the UK Biobank, where they've scanned thousands of participants with their multi-organ MR protocol, and they've extracted a lot of interesting features, for example, structural and functional connectivity in the brain, cardiac and cardiovascular function, as well as various measures of liver health, and all of which I think are quite complementary to what PET can give us. And so I don't see this as sort of a competition, but rather if you can use both modalities, then we can more holistically paint a picture of the whole body. And so with that said, I think the Martinos Center has a very unique environment to take advantage of these new technologies. Using 7TMR and the Connectome scanner, you can probe at high resolution brain anatomy, structural and functional connectivity. And with PET MR, you can assess the neurochemistry. And soon with the high resolution brain PET MR system, maybe we can start probing these smaller nuclei in the brain that we think communicate with the rest of the body. And I feel that total body PET can be this bridge to connect these different modalities, across scales, to link the brain with the rest of the body. Of course, I think most of you know that these first three modalities are Dr. Rosen's 3 great brain observatories. I hope that total body PET can find a place in this list as well. So I'd like to acknowledge everybody at the Explore Molecular Imaging Center, especially my postdoc advisors, Drs. Guobao Wang and Simon Cherry. And in this presentation, I showed a lot of different tracers, patient cohorts, and studies. And this all could not have happened without the great collaboration that we have at the Explore Molecular Imaging Center. So thank you, and I'm happy to answer any questions.